Now, statistically, it should be 25% are homozygous dominant, 50%, so we'll write it out like this. One of them should have this genotype, the two that have this genotype, heterozygous, and one that is homozygous recessive. Is there a case that, let's say you have two parents that are heterozygotes, would it be possible for them to have 4 kids that were big A? Could that happen by chance? Yes, it can happen. This is just showing you statistically what is expected. And it also kind of emphasizes why it was so important that you use a model organism with a lot of progeny. The bigger the progeny set, the closer it tends to be to the expected ratios. So in your notes, I actually want you to have this characteristic genotypic ratio for a monohybrid cross. So you should kind of just know this off the top of your head, because we'll look at cases where we're looking at more than one trait. And it's kind of nice to be able to predict what would the genotypic ratio be. What would the phenotypic ratio be for this? How many of them would have the dominant trait? Yeah, 75%. You can write it like, I don't care how you write it, if you do, how you write that out. 1 out of 4, so 25% would have the recessive trait. So it would be a 3-to-1 ratio. If you ever see 3-to-1 ratio, you can expect that it was probably, the parents were probably both heterozygous. Anybody have questions on just terminology of that? Okay. So when we get back to what he did, he maintained the true breeding lines using self-fertilization. So for self-fertilization to happen, essentially you just have to isolate those plants so that they're not getting fertilized by outside sources. For the cross-pollination, or outcrossing, we call it worms, cross-fertilization, you actually control what the pollen is and where that pollen goes. So in order to do that, you can remove the male, what is it called, stamen? I forget the flower anatomy actually, but you remove the pollen source from one plant, and you can actually use the paintbrush to physically transfer pollen from one plant to another plant. So you are completely controlling the mating in that case. So this is how he would do the F1 cross. He would control which ones were fertilizing which. Okay. So we already talked a little bit about this. I don't think we need to go through the cross. This is just another example using round versus wrinkled. You take true breeding round cross with true breeding wrinkled, you get 100% round. So which one is dominant? Round. And we talked about the enzyme, that was the starch branching enzyme that explains that trait. And then when we take 2 of these, F1, and cross them together, we get the F2 cross, which is this one. Okay? And that would give you the expected ratios, just like we saw here, and 3/4 of them would be expected to be round, 1/4 would be expected to be wrinkled. So through all of these monohybrid crosses that he did, he came across this idea, or kind of formulated this idea, that something is being passed. Because here's what was interesting. When he took the wrinkled and the round and he mixed them together, mated them, the wrinkled trait disappeared. It was gone in this generation. But then if you took these two F1s and crossed them, in the second generation, this trait reappeared. That completely dispelled the myth about blending, right? If you take 2 colors and you mix them together, can you ever get them back to the original 2 colors? So that kind of proved that it wasn't something wasn't being mixed, but there's actually a discrete thing being passed, and it was just being hidden in this F1. So this helped him to come up with this first law, which is called the law of segregation, or the principle of segregation. And that's the idea that something was physically being passed from generation to generation, and it was really the reappearance of the recessive trait that pointed out that, okay, something had to be hidden in that intervening generation. So it was still being passed, but it was being hidden. So how can we explain this reappearance? Well, what Mendel came up with is this idea that these traits, they're discrete traits, they're carried on something. At this time, they still don't have an idea about chromosomes or genes per se, but there's something being carried. And when we look at the parent, so this is the heterozygous parent, when it goes through meiosis, the big R and the little r are going to segregate, right? So what phase of meiosis does this segregation happen? Separation of the two different alleles. Anaphase 1 is when the homologous pairs separate. That's the point when this big R and little r are going to be passed to separate cells, and you have equal likelihood to get both, right? So this is another thing you realize with the math of it all. You were equally likely to produce gametes that had big R and gametes that produced little r. So, you know, the idea that we have in our heads about dominance being like somehow more powerful or something like that, that's not the case here, that you have equal likelihood to get a dominant trait as you do to get the recessive trait. You're going to have 50% of your gametes with big R, 50% with little r. So this is the law of segregation. Does the law itself make sense to everybody? Okay, so then let's go to the dihybrid cross. This is one we probably need maybe a little refresher on how to do a dihybrid cross. Dihybrid, only difference between a monohybrid cross and a dihybrid cross, this has two traits. So we're looking at two different genes. For the dihybrid cross, usually we make the assumption they're on separate chromosomes. And the reason for that is because the way we calculate how many gametes we get, we're assuming that they're actually on different chromosomes and they're independently assorted. So in this case, we're looking at two different traits. One is for round versus wrinkled, the R gene. The other one's for color, yellow versus green. Okay, so here they took a round yellow parent crossed with a green wrinkled parent. They got 100% round yellow. So which of those alleles is dominant? Round and yellow. It's hiding the green color, it's hiding the wrinkled color, or the wrinkled shape. So then to figure out dihybrid, to take those two F1, two of these, and cross them together, we're going to get the F2 cross. So we have to kind of remember, how do you set up a Punnett square where you have two different genes that are involved? Okay, so again, for it to be hybrid, both of the genes have to be hybrid. So it has to be heterozygous for this one, and it has to be heterozygous for this one. So this is one parent, and then we also have another parent just like that one. How do I figure out the gamete possibilities here? Anybody remember a trick? How do you figure that out?
I don't know any trick, we just have all the different possibilities.
All the different possible combinations, yeah. Does anybody remember FOIL? Anybody ever learn?
First out and last.
Oh, Jesus.
You remember this?
First out and last.
First, yeah, so this is a way to figure out what are all the possible combinations. So we can do the firsts. So I could have the big R with the big Y. Then I could have the outsides, big R with the little y, the insides, little r with the big Y, and then the left, little r with little y. You probably remember it when you were, math, when you multiply two parentheses, you use this to figure out. Okay, so we have these possible gametes. We have equal likelihood to get all four. So we have 25% chance to get this combination, 25% chance, etc. So this parent has the same exact gametes, right? So it's going to be big R, big Y, big R, little r, big Y, and little r, little y. So now we're basically going to take these and combine them. That's the idea of fertilization. If this gamete combines with this gamete, we have two big R's and two big Y's. when you write this, one thing I sometimes see is people just write RY, RY, but you need to put the two genes together. So the R's go together, the Y's go together. The other thing is that you always write the capital letter first. So like in this one, you would write big R, and then you write big Y first, and then little y. So I will teach you a shortcut and a way to not have to do this, but you should be familiar with how do you do a dihybrid square. And I think that one of the questions in the genetic problem set, for number 4, they ask you to do a dihybrid cross. So you will have to then draw out your Punnett square and fill out all 16 boxes. And then you figure out what are the phenotypes that go with each of these phenotypes. So you combine all the ones that have similar phenotypes together. Now, if we do that here, how many of these have both of the dominant traits? So in other words, how many are round and yellow? Count. We have 9. So there should be 9 16s that have both dominant traits. How many of the traits just have the round, but they're actually green? Okay, so we have 3 16s that have one dominant trait. How about the other one, that they're wrinkled, but they're yellow? 3. Yeah, 3. And then how many of them are both wrinkled and green? 1. Okay, so in your notes, this is the characteristic dihybrid phenotypic ratio, that you will have 9 with both dominant traits. 3 of them will have one of the dominant traits, but not the other. 3 will have the other one, and then only one is going to have both of the recessive traits. So if you ever see 9 to 3 to 3 to 1, that tells you that the parents were dihybrids. Anybody have any questions or any questions about like how you set up the Punnett square? Okay. All right, so what did he learn from dihybrid process? His big realization from this is that those traits had to be inherited separate from each other. They had to be independently assorting in order to get 25% of each of these possible gametes. That told him, well, they must be on separate chromosomes. So here's a visual to kind of show that. If you had the big R, or the R gene is on the long chromosome, and then this Y gene is on the smaller chromosome, if we have alignment like this during metaphase 1, we would get big R with big Y, and little r with little y. But if it lined up like this, we would get big R with little y, and vice versa, little r with big y. And we would get these with equal likelihood. What would happen if the R and the Y gene had been on the same chromosome? So let's say that instead of this, what if they were linked? Where R was here, Y was here. Anybody know what that would change with the gametes that you would produce? Yeah.
You wouldn't get like a big R with a little y. You would only be able to get like the dominants together and the recessives together.
Yeah, so for the most part that's true. Is there a way for us to get the combination of big R and little y? Crossing over, but you're right that most of the time what you would have is this one and this one, and much less frequently you would see those two. Those would be the crossover. But if you ever had that happen, you would not see 9 to 3 to 3 to 1, right? Because the gametes are not produced at a ratio of 1/4, 1/4, 1/4, 1/4. Okay, does that make sense? So this was his realization. His realization was those chromosomes are on those genes are on separate chromosomes. Okay, any questions on those two laws? Law of segregation, law of independent assortment. All right, so the last thing I'll go over is probability, and then we can stop there, and tomorrow we'll pick up where we left off. So probability, as you can see, genetics is actually just probability. It's really just statistics, right? What's the likelihood you get a certain combination? We can actually solve these genetic problems without drawing all this out, all we have to do is figure out what is the probability of getting certain combinations. So if I ask you, what is the probability, if these are the parents, they're dihybrids, what is the probability of getting this combination? We don't have to do the whole Punnett square to figure that out. All we have to do is figure out what is the likelihood of getting this combination and what is the likelihood of getting that combination. And then we can use this rule called the product rule to figure out the likelihood of having both things happen at the same time. So the two rules that we're going to be using for genetics are product rule and the sum rule. The product rule is when you're figuring out what is the chance that one event happens and simultaneously a second event happens. And then the sum rule we'll use if we want to find out what's the chance that one event happens or a different event happens. So to figure out the probability of both things happening, we multiply the probabilities of the individual events. To figure out the likelihood that one or the other happens, we add the probabilities. So to kind of figure this out, let's do these practice ones first. I think it makes sense before you think about genetics, think about it in kind of like practical terms. So flipping a coin. If you flip a penny, what's the probability of getting 5 heads in a row? How would you figure that out? Which rule would you use to figure that out? Product rule, yeah. And so what would you do?
1/2 to the power of 5.
1/2. Would you say 1/2?
1/2 to the power of 5.
Oh, to the power of 5. I thought you said divided by. So, okay, yes, 1/2 to the power of 5. And the reason for that is that we're saying we're going to do event 1, event 2, event 3, event 4, and event 5. In all of those events, it's 1/2 chance, 1/2 chance, right? So this is how she says, get 1. And so what would be the probability of that? 2, 4, 16, 8, 2, 4, 8, 16, 32, right? This is the likelihood that you get 5 heads in a row. If that happened, would you think the system was rigged? Or can you imagine a time you might be able to get 5 heads in a row?
It wouldn't be likely, it's impossible.
Wouldn't be likely, but it could happen, right? So if you think about it, the likelihood of getting a female versus the male is also 50-50, right? It's depending on whether the father passes the X chromosome or the Y chromosome. Each time it should be 50% chance. But do you know any families that have like 5 boys or 5 girls? It happens, right? So with a small data set, okay, that can happen by chance. If you had 1,000 heads in a row, would you think that there was something wrong, or would you just... All right, so the larger the data set, then you don't expect to see as big of a deviation from the expected. The bigger the number, then you expect it to be closer to 50-50. Okay, so with the person next to you, try to do B and C, and we'll go over those two. And then I think, there's one more concept check, and then we can end class. Let's see if you can calculate B and C.
I haven't cut why, how, wait.
Because the probability you had to.
Oh, yeah.
Was 1 or 2. And the nickel was 1 or 2. And the tails and nickel was 1 or 2.
Because you are hyper specific with.
Yeah, and it's N, so it's gonna be tied, right? And it's here, oh yeah, I see. Oh, okay, and then, what's the probability that it has
So,
one and a tail? Okay.
This one's a tricky.
That's 1/4 chance.
All right, so how do we do
Wait.
B?
Oh.
Here, have a penny and a nickel.
It's in either one is heads.
Mm-hmm.
What is the probability that you get a head with the penny and a tails with the nickel? 1/4. Does everybody agree with that number?
Mm-hmm.
So the chance I get a head with the penny would be
It's gonna
1/2. And then I multiply by the chance I get tails with the nickel, that's oops, sorry, 1/2. And then I multiply them, so I get 1/4. Okay, that one's pretty straightforward. For C, now it says the same thing, you're flipping a penny and a nickel, what's the probability of getting heads with one and a tails with the other? How do you figure that one out? Is there more than one way to get a heads with one and a tails with the other? So what's one way you can get heads with one? We would have, let's see, penny could be heads, and nickel could be tails, that's one combination, what's the other way you could get it?
Penny has tails and tails has heads.
Yeah, penny could be tails, nickel could be heads. So I have two different things, I have to figure out what's the chance I get this, and then what's the chance I get this, and I can have this scenario or this scenario, so then what do I do with this probability now? I would add these two together, so what's the chance I get a heads with penny, tails with the nickel? 1/4, right? I'm getting it from doing this, 1/2 times 1/2, which is 1/4. Sorry, it's probably not a logical way to do math, it's like off the page, but this is also the same, right? Chance I get a tails with penny is 1/2. Chance I get heads with nickel, 1/2, so it's 1/4. So I would add those two together, because it's this situation or this situation, and I get 1/2. So that's how you would approach that one. Whenever there's multiple ways to get something, you figure out all the different ways and you add them all together. All right, to relate this to genetics, and this is actually kind of more similar to the problems that you have for number 2. If you had, now you have parents that have this genotype, big A, little a, big B, little b, big C, little c. Oh, both of them have that, okay? So this is a trihybrid. Well, I certainly wouldn't want you to do an 8x8 Punnett square. That would take a long time, right? So instead of doing that, we can figure out what's the likelihood that you get this combination, little a, from these parents. So you just take each gene individually, and then you're going to multiply the probabilities of each of those all together. So if these are the parents, big A, little a, what's the chance you get little a from these parents? 1/4. And you can always draw a little Punnett square, like if that doesn't make sense initially, you can always draw a little Punnett square to the side, but that would be 1/4. Again, big B, little b, cross with big B, little b. What's the chance of getting that? It's the same probability, right? 1/4. And then big C, little c, big C, little c. It's 1/4. So I'm asking what's the chance of getting this and this and this, so I multiply all of them together, and I would get 1/64. That would have been 1 square on your whole Punnett square. So I want you guys to try the second genotype, big A, little b, big C, and then if we all kind of come to the same number, then I think you have a solid understanding of how to do this.
I think it's like 16.
What? I am not there yet.
You're taking statistics and teaching, right?
And I don't think I'll like it.
Consider this introduction. The big A is 1, 2, right?
Yeah, 1, 2, 3, 2. There it is.
Little B is 1,
1,
4.
4.
Oh, that's big C, big C.
Yeah, that was like, is it gonna be 1,
Maybe there's also
1,
1. Yeah, that's all.
right? I'm trying to get that, and then we can add that.
Yeah, half times a quarter times a half is 2 times 4 times 2, which is 16.
Does anyone have a number they want to throw off there?
1/16?
What did you, 1/16? Is that what you said? Okay, does everyone agree? We can check, put the numbers in. So, these are the parents still, and then now we have to figure out what's the chance of getting a heterozygote. So what should that number be? 1/2, yeah, 1/2, because there's two different ways for me to get that. And then little b is the same, so that's still 1/4. How about big C? 1/4. So, does C have 2 times 4 times 4? So what should that number be? 250, so 1/32. So this should be the number that we get, right? And maybe what you had, I don't know if you had this number, maybe that's the number that was, or I don't know, maybe one of these was different. Does that make sense to everybody, how you could calculate that?
That is so much easier.
It's so much faster, right? Okay, so we can end class there. Right at this point, you have enough information to do page 1. This is pretty fast. And then page 2 is monohybrid. So you can do monohybrid cross and dihybrid cross. You could answer the first 4 questions already. So I would, and it might be good to just check and make sure you understand them. Just check on those. And then for tomorrow, of course, don't forget to read for the genetics discussion